In the ultra-competitive world of sport, what if effort isn't enough? Are there factors that one can't simply overcome when it comes to achieving their dream to be a star? This video will take a peek into the genetics influencing success in sport. Specifically, we will look at the MCT1 gene and how it impacts the body's ability to transport lactate, a critical metabolite during exercise. Welcome to the genetic basis of stuff and things. The skeletal muscle contractions that enable our ability to move and exert force are powered by ATP, which stores energy for use at the cellular level. During aerobic cellular respiration, glucose is broken down into pyruvate, which is then shuttled into the mitochondria and used to produce the majority of ATP. However, during high-intensity exercise, the mitochondria are unable to oxidize all of the available pyruvate. This triggers the reduction of pyruvate into lactate in the cytoplasm via the enzyme lactate dehydrogenase. Historically, lactate accumulation in muscle has been associated with fatigue and pain. Rather than metabolic waste, however, lactate is now seen as a fast and efficient fuel source for cells during exercise that can be metabolized along several pathways. Over time, lactate that is built up in skeletal muscle cells can be transported into the mitochondria and oxidized back into pyruvate. Alternatively, lactate can be transported through the bloodstream to serve as fuel for adjacent skeletal muscle, as well as organs such as the heart and brain. If so, which proteins have the job of transporting this metabolic workhorse? The facilitated transport of lactate across biological membranes is carried out by a family of monocarboxylate transporters. Monocarboxylate transporter 1, or MCT1, is highly expressed in muscle tissue, being responsible for the transport of lactate from the bloodstream into cells, as well as into the mitochondria inside them. This control over the input of skeletal muscle's primary fuel source makes the MCT1 gene a very compelling topic of research with regards to the impact of genetics on performance in sport. A common polymorphism located at coding base 1470 of the MCT1 gene was identified by Merin and his colleagues in 2000, with A and T variants. The transversion from A to T results in a conservative missense mutation, causing a change from glutamic acid to aspartic acid in codon 490. The T allele is the major or most common allele, with a frequency of 0.59, thus making the A allele minor. Cupeiro and his colleagues investigated the influence of the MCT1 A1470T genetic polymorphism on blood lactate accumulation after exercise at various intensities. A sample of 10 men was used. The participants were put into two groups, depending on whether or not they carried at least one T allele. To determine this, researchers performed PCR amplification and gel electrophoresis on genomic DNA extracted from participant blood samples. In the study, the participants completed 3 sessions of circuit weight training at varying levels of intensity, either 60, 70, or 80% of their 15 repetition max, which is the most weight the participants could perform 15 repetitions with. Blood lactate concentration was measured throughout the sessions. This data was then plotted over time, and a linear regression equation was used to determine the rate of lactate accumulation. The results showed that at 60 and 70% intensity, there was no difference in the rate of lactate accumulation between the genetic groups. However, at the highest intensity of 80%, T allele carriers had a much higher rate of lactate accumulation compared to non-carriers. Additionally, T allele carriers had greater maximum lactate concentrations than non-carriers. The researchers concluded that the T allele may impair MCT1 function, reducing lactate transport capability from the bloodstream into muscle cells. This would explain the faster accumulation and greater concentrations of blood lactate during high-intensity exercise. But why did these differences only appear at the highest intensity? The researchers proposed that at lower intensities, the impaired MCT1 transporter is able to handle the amount of lactate being produced. However, the greater amounts of lactate produced during high-intensity exercise overwhelm the impaired transporter, causing lactate buildup within the blood, while the transporter in AA homozygous individuals is able to cope with the increased supply. The researchers argue that the reduced lactate transport capacity could increase muscle fatigue in T allele carriers, thus reducing performance at high intensities. Through their work, Cupeiro's team expanded on Merin initial discovery, showcasing the potential of the polymorphism to affect exercise performance. The results of Cupeiro's initial study implied that individuals carrying the T allele may be at a disadvantage when it comes to achieving success in sport. But what have subsequent studies said on the matter? The Cupeiro team carried out another study 2 years later using a greater sample size and more varied testing. However, they found the TT genotype induced lower blood lactate levels than the AA genotype, a result opposite to that of their 2010 work. Conversely, other studies have found results aligning with their initial paper. Luckily for us, there are studies that have found promising links. Some researchers have associated the A allele with superior high-intensity power and sprint performance, maybe due to a better response to high-intensity training. Other researchers linked the T allele with better lower-intensity endurance performance, possibly due to an improved ability to use lactate as an energy source during prolonged exercise. Nevertheless, there is contradictory evidence against both of these arguments, preventing us from drawing too sure of a conclusion. The unclear nature of research into the A1470T polymorphism makes it hard to apply with the goal of achieving success in sport. One suggestion, however, is to individualize training based on the strengths of one's genotype to reduce muscle fatigue and improve performance. It seems that the MCT1 A1470T polymorphism only plays a small role in success in sport, if anything. No matter which A1470T genotype you have, one thing is clear: you can do it.